Hi, we are from Group 1 AS2111A-1 are going to present cells mini project.
Cellular respiration is a biochemical process in which cells are converting organic molecules into energy in the form of adenosine triphosphate. For task 2, we are going to determine the cellular respiration of yeast by using spectrophotometer. What is redox reaction? Redox reaction are words consist of reduction and oxidation, which are chemical reaction which electrons are transferred between two substances. Catalyst activity is the ability of the enzyme catalase to facilitate the breakdown of hydrogen peroxide.
So moving on to the methodology, in task 1, the objective was to observe redox reactions and compare catalyst activity in living tissues as well as to investigate the effect of temperature on catalyst activity. To begin, 2 cm of potato, apple and salt pits were cut and placed in separate test tube containing 5 ml of 3% hydrogen peroxide. The tissue sample were immersed in solution and after 10 minutes the intensity and the height of foam were produced with recorded. For the second part of task 1, the effect of temperature on catalyst activity was tested. 4 beakers of distilled water were prepared at different temperatures, then 5 grams of liver were added to 5 ml of 3% hydrogen peroxide in each test tube, record the result after 10 minutes. Meanwhile, for task 2 is cellular respiration in yeast. Prepare 2 grams of yeast solution and then 5 ml of 20% of glucose, lactose, maltose and distilled water were poured into different test tubes. Next, 2 ml of yeast suspension and 2 to 3 drops of BTV were added into each test tube. Incubate the sample at 30 degrees Celsius for 30 minutes and measure the absorbance at 595 nanometer using spectrophotometer.
Hi, now I will explain to you the result for experiment 1 in task 1. Catalyst activity can be measured by the decomposition of hydrogen peroxide. In this experiment, hydrogen peroxide undergoes redox reaction where it simultaneously acts as an oxidizing agent and reducing agent. According to the result, soap pits have shown the highest intensity of bubbles produced, which is 5, very fast. It means that it has high catalyst content, making it the most effective catalyst. Meanwhile, apple produced the lowest intensity of bubble and lowest foam height, which indicates lower catalyst content, and potato goes in second place. Meanwhile, for the second task, the reaction rate is highest at 40 Celsius. This proves our hypothesis, which is catalyst is performed best around 37 Celsius. However, if the temperature is too high, the enzyme will be denatured. We can see that in 60 Celsius of distilled water, the height of foam decreased to 0.5 cm. But when the temperature of distilled water is 20 Celsius, the reaction rate is low because the reaction rate reduced to 0.2 cm.
Have 4 test tubes, which is distilled water, 20% glucose, 20% maltose and 20% lactose. And for the lowest average absorbance is 20% glucose followed by 20% maltose and then followed by distilled water and 20% lactose. Why does this happen? Because for 20% glucose it has the lowest because yeast could directly metabolize glucose, and for maltose yeast needs to break down maltose into glucose and then it could metabolize it, and then followed by distilled water. Distilled water has no sugar, so there are no respiration occur. And for lactose, there are only certain yeast that could metabolize lactose, therefore there are no respiration occur.
So the conclusion for first experiment is the experiment study that catalyst activity and yeast respiration. Catalyst in chicken and liver potato broke down the hydrogen peroxide into water and oxygen. It works best at optimal temperature by extreme heat stopping its activity. For yeast respiration, we use a spectrometer. Glucose shows the highest respiration with the lowest absorbance, which is 0.782. Maltose has lower activation, which is 0.975 because it first breaks into glucose. Lactose and water show almost no activity, which is 1.535 and 1.43. Since yeast can use lactose or metabolize it without sugar. Glucose is the most efficient sugar for yeast.